Okay, so my name is Arup Chakraborty, and on behalf of eLife, I'd like to welcome you to this webinar, which is part of the launch of a new section in eLife, the Physics of Living Systems, which incidentally launched starting today. So as you know, in just a few years, eLife has become one of the journals that publishes some of the best work in the life sciences. And there are many reasons for this, and principally, the reason is our review process, I think, which is all done by practicing scientists, and also there is a consultation amongst the reviewers after the paper is reviewed, so that you get one set of comments of essential points that need to be addressed, rather than a whole laundry list of things to do. We are very excited about launching this new section on the physics of living systems. And I'll just say one or two sentences about why. The function of many biological systems is mediated by cooperative dynamic processes, often driven far from equilibrium. And they also often involve many participating components. When a biologist or someone trained in the physical sciences looked at the same problem in this class, they often ask somewhat different questions. And when there is synergy and complementarity between these questions, much progress can accrue. And there is a lot of exciting things happening now in the general field of physics of living systems, and eLife wants to be the home for the best work in this field. So this webinar is part of the efforts to introduce this section, and today we have three leaders in the field who have kindly agreed to share their perspectives. And in no particular order, Boris Shraiman is here from the University of California, Santa Barbara and the Calvi Institute of Theoretical Physics, Siggia from the Rockefeller University, and Aleksandra Walczak from the École Normale in Paris, and myself. So first, each one of these experts will share some of their perspectives and prepared remarks, about 10 minutes each. And after that, we will open it up to questions, and we'll begin with Boris Shraiman. So Boris.
Well, hello. And how do I share my screen? Can you see it? Show my screen. Can you see it now?
Yes. Perfect.
Okay. Well, hello, everybody. I thought I would start just by sort of cutting and pasting the questions that, so the three questions that the panelists were presented with. Now, one being, I guess, question number one, what are some of the key questions that are being or should be? And I think in my version, it will become could be addressed by scientists working in physics of living systems. Question number two, how does our field impact understanding of biological systems and processes? And three, what are some elements or features of valuable paper? And I think what I'm going to do is start with, so permute the order, start with question number two. And at least the way I see it, physics of living systems is really part of a much larger quantitative biology enterprise, which uses quantitative methods and ideas, models to study biological phenomena. And so physics is very much, physics of living systems is very much part of it, and it contributes directly and indirectly. So what I mean by directly. So for example, last week I was at a meeting on mechanobiology in Princeton. It was a relatively small meeting. It was still about 100 people, and half of them were physicists, I'd say, other half biologists. Of course, luckily, there's enough interdisciplinarity emerging in the system that sometimes these boundaries are rather murky. Just exactly who is a physicist and at what stage a physicist becomes a biologist. But in any case, so the subject of the meeting was mechanobiology, and that addresses the cellular processes that generate mechanical, morphological changes in tissues. So, compression or flow or change in shapes of compressed tissue layers, buckling, which generates sort of three-dimensional form. And so cellular processes affect mechanics, but then there's evidence that mechanics affects cellular processes back, that there is feedback that controls important processes such as protein localization, gene expression, ultimately cell division. So, and that is clearly the field where physics plays a very important role, and it is also very clearly integrated into biological mainstream by now, I would say. So another emerging field at an interface involves membraneless organelles, phase-separated liquid bodies that have been discovered inside cells. They come in many different flavors and turn out to be very important for a number of different functions, both signaling and enzymatic functions. So, in any case, both of these examples involve physical phenomena in cell function, and physics is quite central to understanding. But then, of course, there are many more examples on all scales, from atomic to ecological. So there is interesting new work on mechanics of allostery in proteins, and there is interesting old and ongoing work on stability and dynamics in Lotka-Volterra models of ecology. Of course, at this stage, you may say that Lotka-Volterra models of ecology are not exactly a very direct application of physics, and that's, I think, where I transition to indirect part of the story, because in fact, direct and indirect is rather a continuum of which depends a little bit on how narrowly or broadly we want to define physics. And I personally am for a very broad definition of physics. So, statistical physics has been contributing to bioinformatic methods and tools for a long time. Some of the more recent exciting examples is the use of max entropy and direct coupling approximation in sort of bioinformatic approach to studying protein folding. Eric Siggia, for example, some time ago contributed greatly through use of physics, statistical mechanics ideas in the business of transcription factor binding site discovery. Aleksandra perhaps is going to tell us more about dynamics of natural selection, which is another exciting example where a great deal of progress has been made in the last 10 years. And the ideas there unmistakably come from physics, but of course, population genetics and natural selection are not what you would think of as physical processes. Nevertheless, physics ideas apply very usefully. Now, of course, spin glass concepts have been prominent in the theory of learning for some time. This whole field of neurophysics or neural computation has a very interesting broad interface with physics. We can talk for an hour just about that. My favorite toy example of sort of indirect application of physics ideas is the problem of planar cell polarity, right? Epithelial cells polarize. So, for example, they grow hairs in a rather coherently directed way. And in thinking about this effect, the analogy with ferromagnetism immediately comes to a physicist's mind, and it's a productive analogy, even though there are no little magnets in epithelial cells. So many more examples, what I would call applied physics. You may ask, is there fundamental physics? And well, and again, the question is, well, how broadly we want to think about, how broadly we want to define physics. And I would so suggest that, well, physics of living systems can undertake so the study of rather ambitious and broad and general questions. And what better place to start than so the problem of growth and form, right, articulated by Darcy Thompson 100 years ago. But wouldn't it be fun to revisit it now with all the knowledge that we've acquired from developmental biology in the last 100 years? So how does biological development take us from genetic information to geometry, to the morphology, form and shape of multicellular organisms? Even broader and more abstract question of how complex is living matter? So is there some way of characterizing the complexity of the phenotype-genotype relation, or complexity of the mapping of a genotype into the phenotype? How many different phenotypes can be encoded with a fixed number of genes? Is there a sense in which evolution favors simplicity? Are certain phenotypes more complex than others? And related question is, well, can we understand evolvability of phenotypes? How far phenotypically can we go in with a finite number of mutations? So, for example, one can try to connect this to the question, the genes to geometry question. What is the space of shapes available to morphogenetic processes? So all of these are examples of questions that our biology friends would not necessarily be driven to ask, but if it were possible to sharply formulate these questions so that they have falsifiable answers and could drive us towards design of new experiments, that could be a worthwhile exercise. And anyway, so this list continues. I think I'm taking too much time, and I'll just emphasize that so these may look like broad and vague questions, but there is some logic in trying to define these questions. So they have to be vague in the sense of generality and opening new avenues for understanding. They have to be well posed. I already tried to emphasize that. They have to generate informative and falsifiable hypotheses. And of course, hopefully, physics ideas could contribute in answering this question. And I guess move on to this last question of what are the elements and features of valuable paper? Well, no particularly new insight here. So obvious things. Uncover a new mechanism, explain observations, generalize, make predictions, uncover connections between different phenomena. I think that's very important, and that's so in particular theorists could make a contribution. And as we formulate the criteria, I would very much like us to test these criteria by asking whether Hopfield's brilliant 1982 paper on neural networks could pass these criteria and get through and published. It was actually published in PNAS as a contributed article, right? So for some reason, peer review was not trusted with that. So anyway, I'm sure my 10 minutes are up, and I'm done.
Okay, Eric?
Yes, okay, so let me, so I have to accept to share screen. Yes, show my screen, and then I have to hit play. Okay, very good. So am I visible?
Yeah.
Fine, okay, so I have also taken Arup's 3 questions, and my responses are largely concordant with what Boris gave you, so I will probably get done in less than 10 minutes, hopefully. And also, I suppose our performances are examples of convergent evolution, since we didn't collude on these things, but seem to have arrived at similar remarks. So as regards questions, I think the list here is infinite, and I just gave some rather high-level remarks here. Obviously, evolution is the one theory that biologists accept as being a theory, but it somehow is a retrospective one, not a prospective one. And of course, we would like to ask whether there's anything prospectively that can be said about that. Computer science furnishes analogies as to what can actually be predicted about learning systems, and evolution is just another form of learning. So there is hope there for saying at a suitable level of abstraction, of course, not genetic sequence, as to what can be said about evolved systems in some useful predictive way. The issue of genes to embryos was mentioned by Boris. Obviously, our biologist friends now give us immensely large genome-wide data sets of everything, binding to everything in every cell, one cell at a time. With all that information, however, I am fond of remarking that one could not predict whether a given genome turns into a chicken or a sea urchin. Perhaps, and there's infinite numbers of problems along the way from genes to embryos. Perhaps Arup will tell us someday how much of the adaptive immune system is contingent, or whether it's just some crazily constructed ad hoc system, or whether there's some deeper, more functional logic to it. And then, of course, the general rubric of limits on biological performance is well known in the neural context. There are similarly cellular variants of that, and of course, the immune system itself. As regards techniques that are used, I guess Boris was thinking more about proper theory with a capital T, but of course, one should also include under physics of living systems experiments that a biologist would never do, i.e., for example, the very quantitative attention now paid to microbial evolution in the lab. That's going on in a number of places with very quantitative comparison with theory, or a spin-off from Boris's work on evolution of the flu. How does physical living systems impact our understanding? Well, it depends who you ask. I have a rather more jaundiced view of this than Boris. I think our biologist friends would say not at all. We're valued for tech support, but not too much more. Are our colleagues uneducated? Not terribly. one failure of the community is sometimes to spout vague generalizations that say nothing about everything. And on the other hand, sometimes one builds an exhaustively complex model which attempts to say a great deal about something very, very particular with sort of fudged parameters. The physicists, I think, employ a lot of biometaphors. If one takes a rather stringent criterion of, some, shall we say, proper theory giving rise to experiment and then interesting non-trivial concepts, the list, in my view, is rather small of such examples. I mean, Turing patterns and whatnot being one, Berg-Purcell and the influence of noise and small size being another. But I don't think we have a great deal to our collective credit. As regards to elements of a good paper, I would privilege one, that a real problem is addressed, i.e. not something for which you have an answer and then go looking for an experiment or something to provide the answer. To slightly victimize a friend, once upon a time someone published a paper about making stripe patterns in a sheet of bacteria using some synthetic bacterial thing. And the notion was this was supposed to be an example of developmental patterning. Well, I would say not a very good one. And of course, Drosophila embryo is vastly more informative than some synthetic E. coli secreting this and responding to that. So, I mean, of course, it's hard to define abstractly what a real problem is, but something that maybe our biological colleagues would appreciate the answer to. A good paper, I think, in our area would unify diverse areas. I mean, again, Hopfield's kinetic proofreading did that, Dzarzhinsky's theory of non-equilibrium things, maybe information theory, cutting across neural science and also development. And of course, one wants a real prediction in the sense of physics, not just explaining what you put in. And regards techniques and sort of modes of approach, one should not denigrate the useful idealization. I mean, in the case of Turing again, I would argue that it is exceedingly hard to actually show Turing's system is operating in any real system, and is always overlaid with mechanical things and more physically, more force-generated patterning systems. But it's a very useful idealization. It's one of those things which may not be strictly true, but even if false, was very, very revealing. Phenomenology should also be underlined. By this, I mean as a way of getting away from infinitely long lists of genes. One knows from a great deal of biology that phenotypes are rather stable. When you mutate systems, you don't typically get mush, or you don't turn a mouse into a fly, but you do homeotic transformations on bits of the fly or something. So phenotypes are relatively robust and limited, and therefore phenomenology seeks to relate phenotypes and does so in a mathematically compact manner, which requires some sophistication to present, and then, if all goes well, enables quantitative predictions from rather minimal models. Another useful approach, which gives rise to results that have some long-lasting value, is the long history of putting bounds on biological processes, i.e. Geometric optics tells us how accurately we can perceive. Berg-Purcell tells us how accurately we can sense from a few molecules. So those are all calculations that have really hard, everlasting validity. And one should also not in any way denigrate, particularly in this day and age, the potential impact of data collection and inference. I mean, Darcy Thompson being perhaps the best known example, but in recent years, from all the genomic data, people are now sort of looking very widely at when various genetic systems originate phylogenetically, and then get some sense of, for example, when metazoans first arose, how the elements of a vertebrate synapse arose from pre-neural systems and so on. So all of these approaches, I think, can be very valuable and give results of long-lasting value. Obviously, how to assess all these issues makes the job of an editor very hard, and one that I don't really relish undertaking in this area, but a great deal hinges on sort of good taste in editors as well as authors. So thank you. Whoops. I have lost my sound.
Can you hear me?
Yes.
Okay. I actually do have a presentation. I just had the computer crash, so okay, it's not gonna work.
Aleksandra, yeah, you'll be able to press it
yeah.
now.
Sorry? Is it, so can you, okay, I'm on, right?
Yes.
And how do I share my screen?
Did you get a little message right now that's ask if you want to share your screen?
No.
Let's try that again.
Okay, here, yes. But no, I don't have a message with I want to share my screen.
Well, it will come
here,
again.
yeah, okay, good.
Right now.
Good. Yeah. Yeah, good. Okay.
Yes, we can see your screen now.
Can you see my screen?
Yes. Whoops.
Because I can't.
Yeah, yeah,
so
yes, we can see your screen now.
Yeah, so just
okay.
You can switch to the presentation.
hit play on the PowerPoint and you'll be there.
Yeah, okay, so I'm gonna try and I really don't see it. What can you see? You can see Finder windows, or you can see a PowerPoint a presentation.
We can see a lot of windows.
Okay.
We can see a lot of windows open.
Sorry. Maybe I should just start talking because it's not worth it. Okay, so let's not share. Okay, here we go. So this is fine. This is okay.
Good.
Okay.
Yeah, we can see your
so
slide now.
So I also wanted to share some thoughts about the links between physics, biology, and so on, with a rather controversial title, which I admit. And I don't think I'll add anything terribly new to what Eric and Boris said, but I think the question we ask ourselves as physicists is how do living organisms work, with maybe an emphasis on the do and the work. And when they work, they do really a lot of amazing things which we can translate into quantitative questions. So they reproduce. That's evolution that's driven by rare events. When they compute, they integrate information and they integrate signals, they sense, they also integrate signals and transmit signals. When they make decisions, that means these processes are irreversible, so non-equilibrium processes. This is an area of physics that needs a lot of work, and we find a lot of inspiration in biological systems, but we don't really know how to deal with it. And of course, work is all of these things. But when they function with a lot of constraints. So there's physical constraints that come from noise, timescale, energetic costs, molecular costs, and a lot of things. So I think we should all be amazed that the outcome is, as in the example of development, reproducible, but also that it works at all. So I thought of just naming a few examples very briefly of amazing biological systems that we don't understand, that I think speak very strongly to physicists. So one example is this male moth that has only a few days to find a female, because that's all the nutrient resources that it's given, and it has to do it in a turbulent environment of air. And this is a problem that Boris and Massimo Valcasola have worked on a while back, providing some ideas that one way it could do is to make a probabilistic map of the sources. So use observations and combine observations that it updates with some memory and map that it's keeping. And then it maximizes entropy. This is just one idea, one example, but I think the idea of learning the environment and the interactions with the environment, how biological systems deal with it, is a fascinating area that's present in neuroscience, ecology, the immune system, and many other places. So we don't really, I think that's an interesting direction. Another one is memory. So the Hopfield model was already mentioned. So that gives us an idea for long-term memory. I apologize for the French. As I said, I had computer problems. But there's also short-term memory, which is encoded in continuous attractors. So how this questions there about how is this really encoded? How are these continuous attractors encoded in the nervous system? How does it interact with long-term memory, and so on? And again, if we talk about the question of memory, this brings us back to learning, it brings us back to adaptability, and it brings us back not only to neural circuits and behavior in general, but also to a lot of molecular systems that are showing memory and complex systems such as the immune system. And again, as Eric has pointed out, what physicists like to do is on one hand learn from data, and that's what physicists did a long time ago when they come up with abstract theories that they can go and check against data. So I think there is this element of the back and forth that we like to find abstraction, but then we want by learning, and then we want to check it, and I think that's valuable. And here maybe there's something else I'd like to say, which is I think what physicists can also bring, not only physicists, but there's some culture of physics of that, is somewhat informed leaps of faith that seem like crazy ideas, but can give rise to completely new areas. And there's many examples of that in biology, like Bernard's clonal selection theory, which didn't come from nothing. There was experimental evidence, but it's the integration and proposal of something that's completely different. So I mentioned behavior. I think this is an interesting field of neuroscience beyond neurons, where physicists have contributed to understanding how large-scale interactions come from short-range interactions, and beyond sort of this being somewhat times very technical, it does have biological consequences, for example, about how information is transmitted. And then the last thing I want to mention is of course one of my the subjects I find really interesting as a physicist, because evolution is the ultimate rare event system, and there's a lot of questions there about predictability and reproducibility of evolutionary dynamics. What are the limits to prediction? And there has been a lot of progress on this, but there's still a lot of work to do, again, with interactions with the environment and looking at real systems. So this is just an example of the prediction for flu prediction, but can we go further than that? What are the constraints? So I think the main thing is, for me, physics of living system happens at all scales, from the molecular through the cellular organismal to the population level. And I view physics of living systems as a very broad topic, which, as Boris pointed out, is not limited to the things that many people in other fields typically think of as physics. So for me, everything I mentioned and everybody has mentioned is relevant to physics. And I think there's also something that Eric and Boris emphasized, is the idea of reduced descriptions and some phenomenology that can be useful. So I think either way, I don't think I had that much to add on top of Eric and Boris, but that's how I see things.
So as I anticipated, there's not a whole lot that I would have to add that would not simply replicate what's been said before. So let me just say that some of the questions that I had thought about that were important to address, people, between the three of you, you've all mentioned it. In my case, I thought of, one that was mentioned by several about what determines size and shape and form. Another one has also been mentioned, but let me say it a bit differently, is how biological systems process and learn information. And this across scales, from transcription to cell signaling to whole organisms to populations of organisms. And also immunology and virology have also been mentioned, as well as are there new principles to learn that unify different biological phenomena. So let me just say a few things about what I think are great papers, or which also does not add very much more to the discussion. The one thing that I did want to say is that Eric is the only one of us in the panel who had, excuse me if I'm wrong, who has done some experiments. And the fact that we did not have a full-time professional pure experimentalist is not by design, but by availability and choice and so forth. So I think we are very excited about, I'm very excited about papers that develop a new experimental method, for example, that can allow us to interrogate the system in ways that could not be done before. And of course, others where you adapt or extend existing methods, theory, computation, or experiment, to ask new questions about systems that have been studied before, and to provide deep mechanistic insights which make falsifiable predictions, as I think one of you said earlier, and also uncover connections between different parts of biology. Now, sometimes this latter effort can involve rich collaborations between biologists and physicists, and those are often fun to see as well. So other than that, I really, even what I've just said is repetition, so I won't say anything more, but rather turn it over for the last 15 minutes or so if there are any questions and discussion points. So Cora, you will tell us how to do this. And one of the email messages I just got was that Aviv Regev is on the webinar, and perhaps we should start by asking her whether she wants to ask a question or contribute a discussion point.
Yes, of course. So just as it's probably visible on your screens right now, to ask a question, please do type it in the question panel on the right-hand side in the control panel for the GoToWebinar that you should have all on your screens. I'm currently looking for Aviv to try and give her this face, but I can't see her, unfortunately, so I'm not sure if she managed to join us in the end.
She just said that she's listening attentively, but will have to leave for teaching soon. So maybe she left.
Okay. But however, we do have a few questions by now from other participants. I can see that, for example, Sandeep Kumar has been asking, would you like to comment on data-driven modeling? That is, should we start with abstract formulation and then test it with data, or should we start with data and try to infer the principle?
Well,
who wants to go first?
I can take a crack. Now, so of course, very often, we start with the data observation, as a point of departure for building a model, and then ideally, your model will not just sort of compress the data, but will also, if you like, generalize, make predictions, and help design the next experiment. So that's what I think both Aleksandra and Eric were emphasizing as rather essential back and forth between theory and experiment. So amusingly, so this brings up another question of well, if you like, scientific sociology of how these collaborations between experiment and theory, or modeling, as it is typically called in the field, how those are organized. And right, of course, we've seen a lot of theoretical physics types start their own lab operations, or as in the case of Eric Siggia, have sort of very close collaboration with a colleague, which of course is just as good, and drive this back and forth in this way. So it will be quite interesting if some sort of separation of labor, such as exists in physics, where it is actually practiced for experimentalists in the field to read a theory paper and at the very least be driven, motivated to prove that it's wrong. So I don't think we have evolved quite that separation of labor in our little world.
I'd just add, that when it does happen, it's really exciting. I mean, I've had many deep collaborations with my immunologist friends over the years, and I think that models created by experimental puzzles, which can then lead to insights that are also testable, can happen with more frequency, if such collaborations can be built across experts on both sides. So anyway, other people, I just thought I'd add that bit. So Eric or Alex?
The only thing I might add is that the technology in biology allows you to produce lots and lots of data very easily on a genome scale, and it's often produced just because it can be produced. So one should not get sucked into it just because it's there. There are example I mentioned was collections of genome data have been used by now lots of people to sort of infer how systems, organelles, whatever might have evolved, which is cutting across a lot of collections to integrate them. But one doesn't want to fall into the role of data analyst just because the data is there, collected for what willy-nilly purposes.
Alex, you want to add something?
I'm sorry, I got cut off and I missed the last 5 minutes. What was the question?
About data-driven models. Your perspective on data.
Yes, I actually do want to add something. So I think data-driven models are great. And I have the fervor of a convert. I think data is very important and learning from it, but I think we can always go a little bit further and we can try and actually find some more models, learn from data-driven models, not just from data.
Cora, do you want to read the next question?
Yes, sure. So the next question is from Pradeep Bandaru. Obviously, excuse my mispronunciation if it happens. So can physical descriptions of biological phenomena be transformed into design principles for cellular therapeutics, synthetic cells, and new forms of life that are virtually indistinguishable from existing ones?
Incidentally, speaking of collaborators, Pradeep Bandaru and I wrote a paper together in eLife, and he's a pure experimentalist. So go ahead. Who would like to take a crack at that first? Boris?
Well, I don't know. I've been taking lots of cracks. I'm happy to have somebody else take a first crack.
Okay, so I'll try, since I'm a little bit involved with that. It certainly is of interest, sort of following the, the Feynman-esque mantra that if you could build it, you understand it. So we do such things in the domain of human development. And of course, that gets you into trouble with NIH because you're not allowed to build humans. That's illegal, or at least not fundable by the government. But I think to build it to understand it is a useful filter on your understanding. And if you're going to advertise your contributions to regenerative medicine, you better be able to build it to specs. On the other hand, that isn't quite theory, right? You could build it to understand it, but that's not the same as abstracting from it some general cross-system idea, which is yet another challenge.
So given that we have just a few minutes left, perhaps we'll transition over to another question.
Sure. So Greg Stevens is asking, what makes a biological question or system especially amenable to physics? Are some systems or questions better than others?
So I will say what I believe, but and I'm sure we'll hear other perspectives. I'm not sure it makes one system better or worse than the other, but I do think that collective effects, non-equilibrium processes, and the transition also across scales are some of the elements that make a biological question particularly interesting to me, and I suspect that some part of the physics community as well. But I defer to other people's views on this as to add the other people's views.
I don't think there's any system that is bad for physics because physics is every system. So I think it's more that some questions are not, you are not going to make much progress using physics to answer some questions. For example, if your question is, what can I make a list of all the genes or all the pathways in the cell? Go ahead.
Well, so somewhat along these lines, I'd say that it's not just a matter of systems, but let's say physicist and a biochemist looking at the same system will perhaps take a different approach and learn different things. I'll give an example of Terry Hua, who ostensibly is his lab has been studying microbial, bacterial physiology, and I would have perhaps thought that this is a biochemistry question, but interestingly, they have found a very interesting sort of phenomenological approach. And even though, again, we were talking about what's physics and what's not physics, using philosophy of phenomenological modeling, they have managed to discover, if you like, a reduced set of variables that control a lot of bacterial physiology, and ended up with a rather predictive set of models, and actually learned some completely non-trivial biology.
I would agree with all the foregoing. It's only a matter of your imagination that limits you, not the system.
So if there's another question, we'll take that, and then perhaps we'll start thinking about wrapping up very soon.
Okay. We might have time for one or two questions. So the first one would be from Andrea Brown. Boris used mechanism as one of his features of good papers. Mechanism can be quite a loaded word that's open to different interpretations by biologists and physicists. Could the panelists elaborate on how they understand the mechanism and its role in the physics of living systems?
Well, this is a question directly to Dr. Shraiman, so he must answer first.
I guess I'm
although we all use the word mechanism, but
I think we all know what the mechanism means, although it might mean different things to different people, and also different things to the same person at different times in different contexts. So I will just rephrase this a little bit. So very often, some of our friends think of mechanism something chased down in its full sort of reductionist glory to the underlying gene and protein structure, and the active pocket, and all of that is true. I think we have also been using the word phenomenology a great deal. So I would say that the minimal definition of a mechanism is a predictive model. So by the time we understand more about the relation of some important basic variables to formulate a predictive model, we have learned something about the mechanism. I think that's my attempt at an answer.
No, go ahead. Sorry, Boris.
No, I think I've taken my crack at an answer. I'll pass it on.
So maybe if there's one more question, we'll take the next question and then wrap up immediately after that. Otherwise, we'll have other people share their thoughts on what Boris said. So is there another question, Cora?
Sure, yeah. We have plenty of questions, actually. I'm not sure if I don't think we'll be able to cover them. But so, for example, Rui Dilao is asking with a little preamble. In physics, we narrow the questions and we analyze specific problems within some framework, mechanics or TEMOs, etc. In all the interventions you were talking about, broad questions, were all broad questions, somehow more metaphysical. Why was that? Is biology too difficult, or do we not have the right tools?
Well, I don't think that we are thinking only of the broad questions. The broad questions have specific realizations and examples in order for, as everyone said, for the models to be predictive as well. So we are thinking of big, broad questions, but also thinking about specific realizations of them. So, but I let other people speak.
Well, I'm afraid that I was responsible of all the panelists for the metaphysical questions, such as perhaps asking a question of how complex asking a question about complexity of biological phenomena can definitely be perceived as a metaphysical question. But my intent in this was a little bit to stir the pot, because we, of course, as a day job, we mostly work on much narrower and specific quantitative questions relating to particular phenomena. And my intent was to provoke and try to open up the possibility of asking, if you like, bigger questions. But at the same time, I tried to emphasize that the key to formulating a question is narrowing it down so that the answer becomes falsifiable.
In fact, the last talk that I heard Boris give was 2 weeks ago at the KITP, where the major tools were geometry and mechanics, if I remember correctly. So
yeah, indeed.
Eric, you want to add something? Because I think we've lost Alex.
I would pretty much say the same thing. I mean, one can daydream about big questions, but at the end of the day, something substantive has to be done with some system which is predictive and falsifiable and somewhat quantitative and not otherwise obvious. So yeah, so I also was guilty of giving some broad categories, but just to explore the scope of what might be reasonable under physics of living systems.
Yes, and the tools are there, both experimental and theoretical, to answer very concrete biological questions. So it's 12:28. So do you think we should take one more question, Cora, or we should stop now?
It's really up to you. We certainly have more questions. One of them that perhaps might be slightly quicker.
There's a short question. We'll do just one more, and that'll be it.
Okay. How do enthalpy and entropy regulate the biological processes in living systems? That's from Xiaojun Ren.
I actually couldn't hear the first part of the question.
Right. Okay. So how do enthalpy and entropy regulate the biological processes in living systems?
The first word was what? Enthalpy?
Enthalpy.
Okay. I couldn't hear. Okay.
I think it's enthalpy. It's just a little bit misspelled here. Sorry. So it's enthalpy and entropy.
Well, the obvious thing that's very interesting about biological systems is that they are driven far from equilibrium, and therefore they create systems that lower entropy, and that's a very interesting phenomenon. But other than that, I'm not sure. And there are many possibly interesting things to learn from that, from the standpoint of principles, but I'm not sure what else can be said without a concrete example in mind. But maybe Boris and Eric, you have something to say?
But entropy also comes up in the sense of information theory, which is entropy.
Yeah.
And it also then comes up implicitly when you think about learning models or learning statistical models. So it's certainly very present, maybe not in its explicit physical, chemical, thermodynamic incarnation, but certainly in a statistical mechanical context, it's very much alive and underlies all sorts of things that go into models, right? So information is entropy, and that covers a lot of biology.
Yep. Maybe Boris, you can have the last word.
Well, actually, the two of you pretty much covered it, so I don't think I have much to add. Indeed, between so physical chemistry, so because physical chemistry aspect of it is perhaps the least interesting, but the opening in statistical physics and information theory is fascinating.
Indeed, and as I think one of you mentioned in your remarks, I think it was Eric, of how much is going on with max entropy models in diverse fields, ranging from virology and kind of things I've been interested in, various other things, some neuroscience and so on. So, okay, so thank you for all the participants and the panelists, and we are very grateful to you from eLife for participating, and we hope that there will be a wonderful section now in eLife for this sort of activity at Physics of Living Systems. Thanks all.
Thank you.
Thank you.
Bye-bye.
Bye.